Evening, evening, evening for me. Morning, morning, morning for you. Okay, let's get the chat up and running so I can see what you've all got to say to me. Okay, so what are we doing today? A bit of mitosis. I know that I said I wasn't going to be here today, Friday. Well, my flight was cancelled. So I am rebooked on a flight for tomorrow. I actually launched a little Instagram story and poll earlier as to whether you think I'm going to get home or not. So my ticket is for tomorrow. So in theory, I'll be good to do all of these classes, but I don't know whether the flights are going to run. I just don't know. Nobody knows. Simple answers to everything. Nobody knows. So mitosis and microscopes content for today's AS class. Suitability is AAQA, OCR, EdXcel. I'm sure if you do other exam boards, there will be lots of relevant content in here. Check your own specifications if that is if you are another exam board. I'm going to start before the hour just with a little bit of showing you inside of the course. I made a few tiny little updates. Oh, I don't think I published it. That's a little bit annoying. Okay, I'll show you some of the tiny tweaks. In fact, I might be able to do it just quickly whilst we're online. Just ask me some questions if you want, and I'm just going to publish this page so you guys can actually see all my modifications. Okay. No one said anything to me so far in the chat. I'm assuming that you can all see me and hear me just fine. It's looking a-okay for once on the front end just here. And there's, okay. Started streaming 102 seconds ago. Okay, so what I'm going to do just inside here is publish my page here. And I'll preview it. So, okay, you are there. So let's. Google Chrome here. Okay, so you should be seeing inside the Exambulance right here. So I've added this documents and downloads page essentially to the study at home rescue package, the top section there. And inside that, you'll be familiar with the AS schedule and so this has got all our classes on it. We are today at Mitosis and Microscopes, and the documents and downloads page has got each of your specifications in it. So you've got your specs to download. This is some useful documents about the math skills. You've got the cheat sheets and stuff from the information on the command words, breaking them down, AO1, AO2, AO3. In fact, this is the AQA document. I need to probably just remove this because this course is exam specific. So this is what AQA students can say in terms of abbreviations. Like you can say DNA, but you can't say TP for trisphosphate, etc., stuff like that. I've also added in some of the template notes from the AS live class recordings. This is obviously going to be joint for the A-levels. If you're an A-level student, you get all the notes templates down here as well. So they're available for you to download. Obviously, we're going to be adding some more content to that. I've also changed the buttons in here a little bit. So if you're in the AS study schedule page and you click this button, it will take you to the playlist of all the pre-recordings of these sessions that we've done so far this week. If you're on the A-level study page, you click the YouTube button, it will take you to the A-level playlist of all the year 13 biology material that I've been doing so far. Okay, so far so good. Well, okay. Oh, has it? Did you see any of that? I've just got a massive delay on here. Or was it just YouTube loops? Oh, what? Madness. Madness, I tell you. I'll give you okay. Let's try that again. Here inside, you should be seeing, let's go back and check. It's weird because I've got, okay, so what I might just do is collapse all my Chrome windows into, oh, you were seeing the right thing that time. Do you have? I hate not using two monitors. I'm such a 21st century problem, and I'm also wobbling my laptop like nothing else. This time. Okay, so you'll basically see this new documents and downloads on the left-hand side in the Study at Home Exambulance package. You've got the documents that I have produced for you, my cheat sheets and stuff like that. You've got the specifications over here with some useful documents from the exam boards themselves. This is the YouTube button that will take you to the Year 12 playlist if you're on the AS study schedule. So on the AS study schedule, you've got here, this button takes you to the AS playlist. If you're on the A-level schedule, which is just there, you'll take you to the Year 13 material. And yeah, that is basically, and these are the notes templates. So in fact, today's notes template, we've got mitosis and microscopes. If you want to, if you've got the printer right at hand, you can download and print them because I'm going to be starting in just a couple of minutes. But obviously the notes templates are skeletons. There's not very much writing on that page. They just give you some nice structure. I will try and get my notes templates in here in advance. So, you know, although that said, hopefully tomorrow I'm on a plane. So I will be hopefully not able to do any of that tomorrow, but we will see. Okay, I need to stop using I've got my super homeschool sort of, normally I have arms for my camera, separate microphones, and just don't have any of my regular setup yet today. Okay, so microscopes and mitosis. Probably chuck in some questions in there as well. I think let's just go, shall we? There's no point beating around the bush too much. Share the iPad. Try not to wobble the camera too much. Okay. So you're seeing that, or you at least should be. Okay, so mitosis. What's the point? We did a little bit of mitosis versus meiosis the other day. In fact, I'm just going to ask you some questions first of all. Wow, this is confusing as hell. I want to do that, and then I want this over here, like that. I think is best. What's the point of mitosis? Why do mitosis? What are the headline details? How many divisions are there? What happens to the DNA? When does the DNA replicate? These are, okay. Mitosis here. It's cell division. Okay, we're number one, we're at cell division. Perfect. We're dividing cells. What's the purpose of it? Growth and repair. So you're producing replacement cells. You will just, okay, so we're creating two identical cells. Yep, they're identical to one another, and they are identical to the parent unless a mutation took place whilst the copying of the DNA was done, which generally speaking, obviously, for the millions of bases that get read, the error rate is very low, but there are millions and millions of bases, so occasionally errors creep in, and those are mutations. Asexual reproduction, yeah, I mean, typically, if you're an AQA student, you need to know that it's called binary fission, when two prokaryotes are going to bud off from one another, but effectively it's the same process, yes. Okay, when does the DNA replicate? When does the DNA replicate? I've seen prophase in the chat there. I've also seen interphase in the chat there. Okay, so DNA is going to be replicated in interphase. Super common question. Where in cell cycle does DNA replication take place? And people will often think that it's mitosis, but no, the replication takes place in interphase, and that the chromosomes. Okay, let's run through. I'm going to put some of these into words in a minute. You basically need to know 2 bullet points for each of these phases. So we've got interphase, we've got, what have we got? We've got the DNA replicates, and we probably replicate some organelles, because you don't want it, you just want a few extras. So when you split it, you cleave the cell in two in cytokinesis, the cytoplasm dividing, but you're not too short on organelles. So yes, you're right, it does, the DNA does replicate in S phase of interphase. Generally speaking, the questions are only going to say which stage of cell cycle, so the interphase is fine. If it said which phase of interphase does DNA replication take place, then the answer would be S phase. Okay, so I'm going to, I think now is probably a good time to be jumping into some notes. Again, you can get these inside the exambulance. Where do you get the exambulance? You get the exambulance on, go to tailoredtutors.co.uk, and you can sign in. I'll put in, if you're watching the recording, I'll get the, this is just, nothing's quite playing ball today. Okay. Interphase. What do we need to know about interphase? I'm going to label these stages. So we've got interphase. And then the stages of mitosis, we've got prophase. Any good acronyms for PMAT? Metaphase, anaphase, telophase. Obviously important to know that. I'm going to draw my, while I'm in blue, I'm going to draw my cell membrane in blue. I'm just basically going to draw a cell for each of these. Oh, actually, I tend to refer to this as like a double fried egg, basically. And then I'm going to switch to black for my nuclear membrane. Obviously, I'm drawing the nucleus massively bigger than it should be. It's not to scale. If you could look at the nucleus and see inside it, then you'll see a dark spot. What's the dark spot inside the nucleus called? I picked my apples today. It's a nice one. So this is the nucleolus, the dark spot. And basically the DNA, we use just a whole mesh. You can't see the chromosomes at this point. They are not condensed. This is what you would see during interphase. The two defining features, basically the DNA replicates. Maybe I can get in a little bit closer in here if it's going to let me. DNA replicates, and I'm going to say organelles replicate. Basically, that's fine. Yeah, this would be our nucleolus. To be honest, we'd probably have something like that. During prophase, give me 2 bullet points for what happens during prophase. Can I give you a little hint by drawing? Well, okay, so the nucleolus is a tightly wound bunch of the chromatin, all the DNA in its loose, stringy form. So when the chromosomes sort of all detangle from one another and condense into forming into their individual chromosomes, then basically you're taking like a big pile of string, and the nucleolus being like a knotted, dense bit of it, you're unknotting it all and putting it into neat little piles. So it basically unknots itself. So yeah, we could say the chromosomes condense. So that means they become short and fat and they become visible. So all that loose, stringy, silly string kind of crazy pattern that they are all in without much organization, suddenly they start shrinking back on themselves and they form these like juice plumpy X-shaped chromosomes. The nuclear envelope breaks down. That's why I've drawn it as dotted line. And we could say maybe the centrioles move to the poles, spindle fibers start to form. I think that's centrioles move to the poles. We could even add spindle fibers form. I wouldn't say that they attach to the chromosomes yet. So we've got our centrioles here. Centrioles move to the poles. Let's maybe make this a little bit. Oh, I'm going to do thick and thin. So this can be my thin one. And they're in no particular order at this point. So here's I've got 2 homologous pairs. I've got 4 chromosomes. I've got the thick blue and thin blue. I've got thick red and thin red. So again, they're not organized. This is the most difficult to spot from an electron micrograph. Usually you pick out prophase by process of elimination. It's not that clear. It looks like a cell with a kind of a blobby bit. You can't really see whether the envelope is breaking down or not. But you can see that it's not metaphase, that it's not anaphase, and it's not telophase. And therefore, your two unidentifiable ones are either going to be interphase or prophase. And you're unlikely to be able to see the centrioles. Typically, it will say, which stage of mitosis is this? Well, interphase is not a stage of mitosis. Therefore, your odd one out must be prophase at that point. Okay, so whilst I've got my thick pen on, let's do, how do the chromosomes arrange themselves during mitosis? And for a bonus mark, how is it different to meiosis? So question number one, how do the chromosomes arrange during the metaphase of mitosis? And how do the chromosomes arrange during metaphase of meiosis? So they don't pair up, so they're in single file. Like this, we have our centrioles here, and we have our spindle fibers. So we could maybe, I don't know, I'm not going to draw any spindle fibers on the prophase one. I've written it down, the spindle fibers form. And then this, we have this attaching to the centromere. So give me, well, we've basically already got, so we've got, I'm going to use the word double chromosomes, but double chromosome is not a key term. You will not get marks for that. So I'm going to put this in my notes on my video. I put this in brackets and I put it in green. It's not a key term. These are, according to the specification, these are chromosomes. So chromosome, can't. Line up singularly on the equator. Again, this is metaphase. I should probably keep the titles in shot. Okay, so who can answer Benin's question? When do the homologous pairs pair up? Okay, let me catch up on some questions here. We're going a bit off track. So in the comments, no, okay, so this is mitosis, where it may be useful to have the full title in there. Exactly, so meiosis, if I was to just sketch what was happening to these in meiosis, I'll do it in the top up here because I'll leave it there, I think it'll be useful. So again, I've got my cell in blue, well, my nucleus is gone. If this were meiosis, I would have here, it doesn't matter which way, it's independent sort, would tell me it's random which one goes on which side. I think I did it in size, oh, I can't remember what size pen I used, but either way, this way. So in meiosis, the homologous chromosomes would line up side by side. And the first division would split these. So these go in this direction and these go in this direction. Whereas in mitosis, we're going to split it down the center and we're going to get half of one and half of the other. But because our two chromatids, our sister chromatids, are identical to one another, you're going to have, it's going to be exactly the same on each side. Whereas because our homologous chromosomes are the same genes but different alleles, they're not going to be identical on either side. So this is the main difference then between meiosis, metaphase, and this is the main difference. And this is metaphase 1, basically. Okay, so we've got the double chromosomes line up on the equator. We could say the spindle fibers attached to the centromeres. That's probably about it, to be honest, unless anyone else has got anything else to add. Yeah, so just to clarify, mitosis, no independent segregation, no crossing over. It doesn't matter if the chromosome lines up this way or this way. It could be either way up because they're identical. The sister chromatids are identical. Okay, so here we have blue thin one is here. They're being dragged. Imagine hooking a hooked piece of cooked spaghetti down the middle and then pulling it through gel. Well, it's going to come first. The centromere is attached here, and the rest of it's just going to trail behind because the cytoplasm is like kind of thick, right? It's just going to, and the chromosome is not rigid. It's just floppy like a cooked spaghetti noodle. So it will just like, these will drag behind. So if you ever get asked to draw them, there is kind of a requirement that they ask you to draw them in a V shape like this. We could say the centromere splits as our third point if we wanted a third bullet point. Basically 2 bullet points is all you ever need. So that's how you draw your anaphase. The spindle fibers, what's happening to the spindle fibers? We can say the spindle fibers contract. The chromatids, sister chromatids are pulled to opposite poles. If you wanted to, I think it would be an acceptable point to say the centromeres split as well. Again, you don't need a huge amount of detail on that. Telophase, why I call it the double fried egg is because it looks like fried eggs that are just merged in with one another. These are obviously going to have one on the left, one on the right. I say the nuclear membrane breaks down. No, reforms, what am I talking about? And then we've got cytokinesis, which I'm actually going to write in red because it is a good key term, cytokinesis, which is the when the cytoplasm divides. What is cleaves, but I'm going to say divides to form 2 cells. What do we have inside them? Well, we've got basically one thin red in each, one thin blue in each. We've got one thick blue in each, and one thick red in each. And obviously after this, the DNA is going to return to looking much like it was during interphase. It's going to decondense, it's going to unwind and go loose and look like that mass of cooked spaghetti again. Lots of spaghetti references coming up today. And then they're going to return to what it looks like in interphase. What part of the drawing is the nuclear membrane? The nuclear membrane is, oh, that's the black. It should be in black. This is the nuclear membrane right here, or nuclear envelope. And it's the same, we've got it here as well. It's the nuclear envelope. It's breaking down in prophase and then it's missing for metaphase and anaphase, it's gone. Okay, I was going to draw a little diagram down here just to clarify some terms in that. During interphase, yeah, maybe I can do it to match. Well, if we could see the DNA during interphase, we'd have one skinny blue chromosome and it would be singular like this, and then we'd have one fat, I'm just going to draw it, one fat chromosome, if we could see them. It's obviously is not visible during interphase stage. And then we'd have one skinny red and we'd have one thick red as well. So this would have one of each homologous pairs. So it's diploid like this. Then as we move on to late interphase, everything's going to double. So we say it's basically 2 times 2n, and we end up with a fat red one that's X-shaped, a skinny X-shaped one that's red. And I'm just doing thick and thin just to denote the homologous pairs. Obviously, they're not actually thick and thin. And so it's still got both homologous pairs present. So we say that it's diploid, or 2n, and it's each individual chromosome has doubled up. And so I personally call that a double chromosome, these X-shaped ones. X-shaped is probably actually a better description to use in the exam than a double chromosome, because double chromosome doesn't mean anything. According to the exam, if we were to draw, this is a chromosome. This is also a chromosome. And then if you are interested, then this is the centromere here. These are sister chromatids, and these are genetically identical, 100% the same genes and the same alleles. So when they divide to opposite poles, one half going to one side in anaphase, one half going to the other half in anaphase, they are identical. So you end up with identical offspring, or the daughter cells. This is a chromosome. This is, I mean, you can call it a chromatid, probably, but really it's a chromatid. Sister chromatids. And these are identical. This is a chromosome. And this is also a chromosome. One of the arms of a double or X-shaped chromosome is a chromatid. I mean, the only time I think it would be that valid to call this guy a chromatid is here they're 2 sister chromatids attached by a centromere. Here they are formally sister chromatids that used to be attached by a centromere. At this point, this is a chromosome. Really, this is a chromosome, because this is, again, this is about to go back to be its normal cell, right? This is these visible chromosomes are about, this is what the cell normally is. This is the diploid, this is the DNA as the cell wants it to function. This is how DNA usually is. It's only ever got these doublings up and forming multiple sister chromatids. So this really is, these are chromosomes. These are the chromosomes that we kind of functionally want. We double them up. We can only see them when they're in their double form, which is a little bit inconvenient when it comes to actually like learning this, because everyone has the impression that this is what chromosomes look like all the time, because that's the only time we can see them. This is what chromosomes would look like if they were condensed and you could see them, but they generally, when you look at a nucleus, you actually see something like this, and you can't actually see the chromosome. So these in the telophase, these are chromosomes. These are not chromatids at this point. I mean, you may get marks for calling them chromatids because they formerly were part of a chromosome and they were 2 sister chromatids at that time. Does that kind of make sense? Like, this really should be described as a chromosome, and this is a chromosome as well, formed of 2 sister chromatids. I would say that they are X-shaped. I think that will give you the marks if you were just determining it. I call them double chromosomes because I find it's easy to differentiate. You probably would get the marks for referring to this as a chromatid, but depending on where and when you're referring to it as. Okay. Let's delve into microscopes a little bit, because I've spent quite a lot of time yapping about that. And then we'll do, if we've got time, we'll jump in with some questions. So maybe do a scale bar magnification question, which students are All the examples introduce that in the new spec, and they are a bit more tricky than the old versions of the questions. Let me just pull up my notes here. Okay, so this is again OCR students, laser scanning confocal microscope, not on here because it's only OCR students do that. I'm keeping these sessions useful to all. How much do Edexcel, any Edexcel students here? How much do you do on microscopes? I'm just trying to think on all the details that you need to know in terms of factual recall from microscopes. Let me just double check. I've just got the spec open here. I've got one of my a billion tabs going on here. Eyepiece graticule. Okay, I can probably find a question on that. It's not the one I prepared, but I can probably find a question on that. Microscope. That's bloody tiny. Like 20% resolution. Be able to recognize the organelles from a microscope. Identify vessels from a light microscope. Use of a light microscope. Okay, in which case, Edexcel students, earmuffs, I'm going to rifle through this super quick, and I'm not going to hang around on it. I will do most of my questions. You do need to be able to do some magnification stuff or some eyepiece practical stuff. I did one with my live class the other week, and it's a tricky one. Okay, you are seeing my screen already. So again, this is light microscopes or optical microscopes. They're easy to use. They're cheap. And they form a color image. So these are the advantages, basically. Disadvantages of using a light microscope is that they have a much lower resolution than electron microscopes. You can't see organelles, basically. You can only see whole cells and tissues. Advantages of a scanning electron microscope. Well, you have basically much higher magnification resolution than a light microscope. Resolution. It really should be optical. Because the wavelength of electrons are smaller than the wavelength of light. You can just say, if you asked why, the wavelength of E minus is my shorthand for electrons, is shorter than the wavelength of light. Another benefit, it forms a 3D image. So basically you're bouncing electrons off the outside and they're reflecting, and the detector is over here. So your source of electrons, you're just firing a scatter beam and they're all going to reflect off. And it's obviously, you're going to see like caverns. It's a bit like, I'm going to say LiDAR, but you probably don't know what LiDAR is. You also don't need a thin specimen, but I'll come on to that in a second. Well, yeah, okay, let's do the benefits of the TEM whilst I'm in green. It's going to save me time. So it's the best magnification and resolution. So forgive my shorthand. I'm just going, you can, so better than scanning as well. And it allows internal structures to be seen. Organelles, for example, anything inside. This is AQA students would be expected to know this, yes. Just not Edexcel. I will put on here. You don't need to know. Disadvantage is it's a lower magnification and resolution than the transmission electron microscope, you can't see internal structures. So you can see how we can sort of pair these things off. Can't see internal structures because we're just scanning the electrons off the outside. Just you're observing the outside, which is why it's 3D. With a transmission electron microscope, what we're doing is basically taking a super thin slice and you're shining a beam of electrons through it. In fact, it probably would just go straight through like that, and your detector is here, but it needs to be uber thin for the electrons to pass through. Whereas with a scanning, you're bouncing electrons off here, and you have your detector here. I don't know why I've drawn. And your electrons are moving like this, which is why you can see 3D structures. You don't need a thin specimen, but you can't see inside. Disadvantage of transmission electron microscope, where you need a very, you can't look at living cells because you have to make a slice through the cell. You really need to use it in a vacuum. Doesn't form a color image is probably next does. Scanning electron microscopes are not color images either, where you can stain them afterwards, but light microscope forms a color image. Transmission goes through, exactly. You need a very thin slice. So preparing the slide is much more difficult. This can create things called artifacts. Basically, by preparing the slide, you can shred membranes, and sometimes you're looking at these shredded membranes and thinking, oh, I wonder what organelle that is. But obviously, before you slice the cell, it wasn't there. You can't really use SEMs on live microscopes. I mean, like, they just wouldn't stay still. Maybe if you knock them out. Generally speaking, no, I don't think you could. I mean, short electron microscope technology is improving all the time. Do I want to add anything else here? Specimens. Specimens need to be in a vacuum. Basically, you suck all the air out so they don't intercept any of the electrons. You want the least amount of interference in terms of, I mean, air might seem empty, but there's ions and molecules all over the place that would interrupt those electrons. Okay, let's go and have a look at this class. So this is a scale. I can maybe drag up an electron. We've got 10 minutes. Do you want me to do an eyepiece graticule question? Is that what you prefer than doing a scale bar magnification question? This is for everybody. Edexcel students, you can take your earmuffs off again. Okay, in which case, you are going to have to ask me some questions because I'm going to have to find one. The good news is, it's what I do. I know exactly which one I'm looking for. It's just the case I'll have to import it onto my iPad, so that shouldn't be too tricky. Okay. That one was super easy. I want to find you the difficult one. Yeah. That's the image. That's the question. Oh, making a mess of screenshotting this. Get all the information in there. Okay, we're good. Now let's send it to myself. Okay, we're up and running. Select a pen and then I can reshare. Okay. Can I show you the previous ones for those of us who want to pause it and try? The other questions, do you mean there? Okay, let's do this question first. Question analysis. Let's zoom in a little bit. So the student looked at a slide containing an onion root tip. Cells under a light microscope. Here they've called it a light microscope, not an optical microscope. Is that big enough? I can make it a bit bigger. Let's just do this. In order to identify some different cells in mitosis, here shows a diagram of what they observe. So it's mitosis. Okay, so we could be asked to identify which stage we're at in each of these. Okay. But that's not the question here, but what have we got to do? The student used an eyepiece graticule, which is shown here. The student calibrated. So the eyepiece graticule, what a lot of students don't, you've got your microscope, which is going to look, it's going to be on legs like this. And this is, you're going to be looking, your eye is going to be looking down through here. And in here, you have a chamber, or you have your eyepiece graticule, which has got your markings on it. It's got graduations on it, or markings on it. And then underneath here, you've got your lens, which is doing the magnification. And normally you can rotate them around. So this might be at ×10 magnification. But your eyepiece graticule is before the lens. And then what you would put underneath here is basically a ruler. So at that particular magnification, you're going to view this, and you are then going to see how many of my eyepiece graticule units make up, and this is going to have micrometers on it. So it's usually called a stage micrometer. You could use a ruler of, as long as it was okay. What was the question there, Henry? Doesn't help because you know how to get the answer anyway. What answer? So the eyepiece graticule is basically, it's in the eyepiece, as the name suggests, which is the top section of the microscope up the top here. And it's, the most important thing is that the eyepiece graticule is before the lens, which is this bit here. And then your stage micrometer goes on the stage, which is down underneath. So as you change the magnification, you're going to basically, if you increase the magnification, you're going to zoom in on the stage micrometer. The stage micrometer is going to get larger. However, the graticules in the eyepiece are not going to change because they're above that length. Okay, so let's go back to this. They calibrated the eyepiece graticule for carrying out the root tip squash. He observed that 20 micrometers measured 2.35 divisions on the graticule. Now, my first question to you, I think this question is naughty, to be perfectly honest. Do you think, what is a division? That's my question to you, as best you can. What is a division? I'll give you sort of options. Do you think it is, do you think this is a division, option A? Or do you think this is a division, option B? A or B? I just want it. What do you think? To be honest, when I did this question, Not sure, exactly. OCR, sort your life out. Anyone writing OCR questions, what are you doing? Like, this is massive sense of confusion here. And there's no allowance in the mark scheme for like what are you doing, OCR? I can tell you, the only thing that they might suggest that it is a little bit unrealistic to do I can't even draw on it, it's that small. 1, 2, and then 0.35 of one of these little ones is going to be a little bit tricky, right? So that is the only clue we've got here, that the answer that they are considering a division is one of these. But shocking question writing, in my opinion. That's not what questions should be testing you on. The setup of the question needs to be clear. What they're examining you on here is your ability to do the calculation, not to understand what they mean by division when it's not clear. So poor question writing, in my opinion. I mean, whether they're actually trying to test you to see that 0.35 means that it's in reality it's too small for you to do that, but I don't really think that's, if the questions are going to be that obtuse, I'd say I'd just use swear words at them, to be honest. That's what I would do. Okay, so let's highlight this again because that's important. So we need to know basically per division, how much is a division in micrometers. So we know that 20 micrometers is equal to 2.35 divs. Yeah, so we're going to divide this by 2.35 to give us 1 division. What is this? Can someone tell me what that is on the calculator, please? 20 divided by 2.35. In fact, I'll do it. 1 division equals 8.51 micrometers. I'm going to tell you that this is a division, because otherwise, I did this in my live class and we ran in circles. Okay, so 8.51 micrometers. So now we know here to here is 8.51. Now what we need to do is use a ruler, and what do we actually have to do to calculate the diameter of the nucleus in cell C? So we've got cell C. So we then use your real physical ruler in the exam to measure this in, let's say, millimeters. What I can do though, we don't have to do it in millimeters here. I can take a line, and I can take a line, let's do it in a color that's going to stand out a little bit more than blue. Let's go back to, that's a little bit short. Okay, so we can take that. So you just measure that distance, and you're going to take it down here and then apply it. No. Just, well. Okay, so then you're going to say, okay, so this is what? I'd say there. How many divisions? So like, what's the final equation that we need to do? What's the final equation? We're most of the way there now. Gonna let you just sit with that one for a second. How many eyepiece graticle units have I got? I want someone else to, I can see, oh, none of you are falling into the trap. Because you've all done this question before, probably because your teachers made you do it because so many students got this wrong. Okay, so we are 1.3. This is not 0 right here. This is not 0. This is 1. This is 1.1, this is 1.2, this is 1.3. So most students here gave this answer as 2.3. Understandably, in fact, in my exam guide, I did this wrong. I just looked at this and was like, okay, 2.3. And then obviously my calculation is wrong because this is not 0. This is 1, and then this is 1.1, 1.2, 1.3. So well done, all of you who are on top of it over there. So we know that 1.3 units. And what do we have to do? We want to times them together, basically. 1.3, because each one is worth 8.5. And what do we get if we multiply them together? What is the answer? 11.063 micrometers. How many significant figures would I give this to? Oh, 11.91. I mean, it's probably because of the way that, why 11.91? Maybe you've done your numbers wrong. I didn't key any of that into the calculator. Yeah, maybe it's 1.4. It might just be, let's just get in a little bit closer in here and see whether my line was actually, how accurate my line was. I'd say that's pretty good over there. Let's get rid of this deleting this here. Yeah, I say it probably is more like 1.4, to be honest with you. I think that is if we were being really careful and looking at that, we'd say 1.4. So what do we get if we substitute that out with 1.4? Times 1.4, and we get 11.91. We get the right answer, 914. But yeah, we've only, in the question, we had 2 decimal places here. I think 2 decimal places is sensible. Why isn't it rounded to 3 sig fig? Well, we've got all the marking guidance down here, so let's just see what the marking guidance says. Oh, and also units. So 20 divided by 2.35, 8.51, that's a mark, if you get that right. Or you get the final answer right without the unit. Seeing as anywhere between 1.3 and 1.5 graphical units. Okay, so it will allow a range of answers then, except in the range of this. So it would allow. Okay, I think that is. Any takeaways? Anything that you specifically picked up there that you didn't know before? Or again, I'm here to basically help you as best I can. All the full content, if you're looking to go into all the more detail, if you're an Excel student, you want to really delve into what the differences are with the microscopes in your course, go check out the courses. Those little details in the question, that little scale bar. Yeah, so really getting to grips with what the question is giving you, and really scrutinizing that is really important. How come it's 1.3 and not 2.3? Because the scale doesn't start at 0. The scale started at 1 and went to 2. So it went the distance, the diameter was the equivalent of 1 to 2.4. So, but it already started at 1, so we had to subtract that 1. So it was actually the distance, the diameter of the nucleus was equivalent to 1.4 divisions. Okay, sweet. The recording will be in the playlist that you're finding the playlists. You've got all the playlists. You can find the playlist from the Exambulance. You can get the Exambulance. Which other question? The scale question. The scale bar question. I think you're talking about this one. Okay, so here we've got two different scale bars. We've got a bacteriophage, which is a virus. Yeah, this is in nanometers, and this is a bacterium, which is a bacterium. And this is in micrometers, so our scales are not the same here. Use the scales in diagram to calculate how many times longer is the bacterium than the bacteriophage. So basically, how long is this in micrometers? What am I doing? And how long is this in micrometers? Or you can do this one in nanometers and this one in nanometers, but you have to do the conversion. So the question is, how many times longer is it? You'd have to, it's going to be a bit tricky to do the measurements on the screen. You can probably figure it out. Okay, I'm going to take a break. I'll see you in 6 minutes for the A-level start. What are we doing in that class? We are doing, if I look ahead at my stuff, a bit of, we're going to basically finish off that biodiversity, a bit of speciation, and look at some questions on that, maybe loop back and just complete that section a little bit better, I think, is what we're going to do. In fact, I need to stop the live stream from here. See you soon. See you on Monday, I hope.